All right, one last topic for acids and bases. And we've been through all of the Ka calculations for the monoprotic acids and bases. We went through the titrations, we went through buffers and all that sort of good stuff. Now we get our last topic, and that deals with titration curves. And it incorporates some of those things that we've been talking about. And these are done in a laboratory situation to track the pH of a titration solution. Sometimes it's important to know exactly when a particular pH is reached for a titration. So in a titration curve, you're monitoring the pH of the solution that's in the flask down below versus the volume of the base. Now there are other kinds of titration. Sometimes it's a precipitation titration or whatever. They all have curves that are similar to this. In this case, it's an acid-base titration, so we're monitoring volume of base versus the pH. And there's two primary types of titration curves. The first one is for strong acid, strong base titrations. And so in these scenarios, we're going to start with the acid in the flask and the base in the burette. The base is the titrant. And so when we work these out and kind of see what the titration curve is going to look like, our plot from this curve, if the acid is in the flask along with the pH electrode, the pH is going to be low to start with. So we're monitoring pH on the y-axis versus volume of base. And what we observe for these, since it's a strong acid at the start, the pH is going to be low. And as you titrate, the pH very slowly goes up because there's still a lot of strong acid left. And then as that acid gets consumed and we reach the equivalence point, the pH jumps because the acid is gone. And now we have an NaOH solution or some sort of base solution. And so on a curve like this, we have some interesting points, or at least one or two anyway. The spot right in the middle of the jump. That spot right there is our equivalence volume. And so that point is the stoichiometric stopping point. That's the most important point of the curve. We also have pre-VEQ and post-VEQ. Now we're not going to do any calculating on these curves. I just want you to see what they look like. Pre-VEQ, there's still acid left, so the pH is governed by how much acid is left. Post-VEQ, the pH is governed by how much base has been put in excess. At VEQ, for a strong acid, strong base, ideally the pH is 7.0. Now our other type of titration curve is for weak acid, strong base. It's still the same setup. pH on the y-axis versus volume of base. Now for a weak acid situation, the pHs tend to be a little bit higher at the start because they're weak acids, right? So we start out a little bit higher on our plot. The pH that you start out at, you can calculate with a Ka calculation, right? But we start out at a particular pH, and there's a slight jump at the start of the titration, and then it kind of levels off, but it's not flat like the strong acid, strong base was. It's relatively flat, but the slope is higher. Because it's a weak acid, the pH is going to be changing more. And then as that acid runs out, we get our jump. We get the same looking tail at the end. So the center of the jump is still VEQ. This time, though, the pH is governed by Kb for the conjugate base. Again, we're not going to do any calculating, but that's where it comes from. Post-VEQ, it still works like the other one. Post-VEQ, it's a dilute NaOH solution, so you could calculate the pH based on how much NaOH has been added in excess. Pre-VEQ, though, it's fundamentally different from the other curve. The slope is higher. This is called the buffer region. Because that's what you've established. As you start to titrate the acid, you generate some of the conjugate base, and you get a buffer. which is why it's flat in the middle here. It doesn't just keep going up all the way till the end point. We create a buffer. And in this buffer region, we can calculate the pH based on the Henderson-Hasselbalch equation, if we were going to do that. We're not going to do that. But that's how these titration curves work when you see them in lab. Laboratory instructors may ask you to do a titration curve and do some calculating based on those spots. Okay? So, good luck.